In this video, we'll be discussing a few different topics that are all related to the nucleic acid segment of the course. There's just a few different topics that we haven't had a chance to get to yet that I'm going to be covering here. So to start off, last time I had you watch videos describing several different techniques that we commonly use in the biochemistry lab, including molecular cloning. And the main idea with molecular cloning is that we're taking a gene that we're interested in and we're putting it into a different organism, in most cases a bacteria cell. And so I wanted to kind of wrap that segment up by summarizing some of the key uses for recombinant DNA, which is what we're doing during this cloning process. So one of the biggest, most common uses for recombinant DNA is to express protein. So we can insert a gene into a plasmid, transform that plasmid into bacteria, grow up the bacteria, and have the bacteria make that protein for us. This allows us to get really large quantities of proteins that we may not be able to get enough of to do experiments with under normal circumstances if we were relying on their natural abundance in the cell. All right, so that's a really important use of recombinant DNA. We can also use it once we have those plasmids for site-directed mutagenesis studies. So we can take that gene and introduce a very targeted mutation into it to see how changing that amino acid would affect that protein function. Right. Recombinant DNA is also the idea behind transgenic organisms, where we're transferring a gene from one organism into another. And it's also the main idea behind gene therapy. So recombinant DNA techniques can actually be used to treat genetic conditions, like they've had some success with severe combined immunodeficiency disease. So that's also known as SCID. And basically, they took bone marrow cells from a patient, they incubated it with a vector containing the unmutated version of that gene that's mutated. The bone marrow cells took that up, they transferred the bone marrow cells back into the patient, and that was an effective treatment for the disease. However, there's still some limitations in terms of using the technology that way, but there does seem to be some promise in using it for cancer treatments, especially immunotherapies. All right, so that's kind of wrapping up our segment on biotechnology. So let's move on to a few other ideas. So the first idea that I wanted to touch on is DNA denaturation. And we mentioned this during the video on PCR. So what DNA denaturation is basically that high temperatures will disrupt the weak interactions holding the DNA together. So the hydrogen bonds and the base stacking interactions. And when those interactions are disrupted, the strands come apart, they separate, and you end up with denatured DNA. And then when the temperature returns to the optimal range, the two single strands will rapidly reanneal in one step as long as they are still attached in the double helix at some point in the DNA. As long as they have that attachment point, basically when you cool the temperature back down, it's just like zipping up a zipper. They're just going to snap into place one after the other very, very rapidly. Okay, so that's kind of the idea of what's going on with denaturation. An interesting thing is that if you have a high number of guanine cytosine base pairs, your denaturation temperature is going to have to be higher. And this is related to guanine and cytosine. That base pair has 3 hydrogen bonds that form. Right, so there are stronger interactions. So if you have more of those, you need to input more energy from your environment into the DNA to get those interactions to break. So that causes that DNA to have a higher melting point. And so we can actually use the melting point that we measure to estimate the base composition. So the way that we do that is that there's this effect that happens when we put the nitrogenous bases in the double helix. It's called the hypochromic effect. And what that is that when you stack those bases on top of each other, when you have that pi stacking, or base stacking interaction, that actually limits how much UV light the bases in the double helix can absorb. So in the double helix, bases are going to absorb less UV light than free nucleotides would at the same concentration. And that's called the hypochromic effect. Whereas denatured DNA will absorb more UV light because now the strands have come apart, you've broken apart those interactions, and the bases can absorb UV light more like they would if they were free nucleotides. This is called the hyperchromic effect. So we have hyper is more, hypo is less. And we can use this to determine the melting temperature to help us determine our base composition of our DNA by monitoring UV absorbance, because it's going to change as we denature the DNA. So how we do that is we start out, I'm going to make a graph, and on the y-axis I'm going to put absorbance, and I'm going to put temperature, and let's just say in Celsius on X. All right, so we're going to start out with low temperature. So most of our DNA is going to be in the double helix. We're going to have very low absorbance. But then as we increase the temperature, we're going to start to denature some of that DNA. Our absorbance is going to go up until eventually all of our DNA is denatured and we have this leveling off point at our highest possible absorbance value. All right, so this up here, this is our denatured DNA. This is our double helix. So we can actually watch this transition happen by monitoring the UV light absorbance. What we can get from this is that this midpoint here, this is your TM, or your melting temperature. And that is the temperature where half of the DNA molecules are denatured. And we can use that to determine our base composition of our DNA. Right, so that's DNA denaturation. So now let's talk a little bit about mutations. So we've been talking a lot about how DNA is this very stable molecule that needs to last a long time so it can be passed on to the next generation. But the way we've been talking about it, we've been kind of suggesting that DNA is this perfectly unchanging molecule that nothing ever happens to. But that's not actually the case. Mutations in the DNA can occur, and they can occur from simple errors during DNA replication, so putting the wrong base in the wrong place during replication, or they can occur from DNA damage, and there's a lot of ways that DNA damage can occur. So let's just start with, what is a mutation? A mutation is any alteration in the DNA structure that produces a permanent change in the encoded genetic information, right? And like I said, this can happen from a mistake during replication. It's rare, but it does happen. Eukaryotic DNA polymerase has an error rate between 10 to the minus 6 and 10 to the minus 7th. So it doesn't happen a lot, but it is possible that the polymerase can insert the wrong base during replication, just by mistake. Mutations can also occur when the DNA is damaged. And DNA damage can happen spontaneously, or it can be induced by something like UV radiation or chemical agents. So an example of spontaneous DNA damage that can happen is what I'm showing over here. So I want to focus on the cytosine and uracil section. So something that happens just spontaneously in the cell, actually relatively frequently as these things go, is that cytosine will deaminate. And what that means is that it's going to lose its amine and its amino group, and it's going to become a carbonyl, all right? And so when that happens, you end up taking cytosine and converting it to uracil. So this is a big explanation for why DNA doesn't use uracil, all right? Because if this happens in the DNA has no way of knowing if that uracil is actually uracil or if it's cytosine, all right? So that's why we use thymine, because that provides us with a level of proofreading. So we know that thymine is supposed to be there, so if we see a uracil, that means that it was probably originally a cytosine, and that's a mistake that needs to be repaired. All right, why is this such a big deal? Well, if I had a cytosine deaminate to uracil and I let that replicate, what's going to happen is that now we're going to turn that into an adenine thymine base pair after many rounds of replication, instead of the guanine cytosine base pair that it should have been. All right, so this is going to lead to an actual change in the DNA as the DNA is replicated. All right, so we don't want this. DNA uses thymine for this reason, to proofread, to prevent this from happening. Right? So that's spontaneous DNA damage, but DNA can also be damaged by chemical agents or by ionizing radiation. So two of the most common ways that DNA can be damaged by a chemical agent are deaminating agents. So they induce the types of reactions that we just talked about, where an amino group is converted to a carbonyl. Right? We can also very commonly see alkylating agents at play. And what those do is they are going to add an alkane, so basically just a hydrocarbon, to another group. So in this case, we're looking at this guanine that has been methylated at this oxygen. All right? And what this does now is that this oxygen was supposed to be a carbonyl, and it was supposed to be involved in a hydrogen bond with cytosine. So now this guanine can't make that hydrogen bond with cytosine anymore. So what's going to happen is that instead of base pairing with cytosine, it's going to base pair with thymine. All right, so this is another thing just like what we just talked about. So now we have a GT. Eventually, over many generations, this T is going to end up making some of the future replications of the DNA into adenine thymine base pairs instead of the guanine cytosine base pair that we should have had. All right. And so this is how DNA damage can propagate through generations and cause actual changes to the genetic information. Mutations can also occur through ionizing radiation. So that typically forms pyrimidine dimers. And you'll typically see that as thymine dimers, where a thymine nucleotide will end up covalently linked to a neighboring thymine residue. So you end up with two thymines that are bonded to each other. All right, that's one thing that can occur. That typically occurs, and that's one of the things that typically occurs when you have UV light radiation. All right, and then probably the biggest source of DNA damage is oxidative damage, and in particular, damage caused by hydroxyl free radicals. All right, very reactive. They interact with the bases. Mutations happen. All right. So that's mutations, talking about kind of some of the main ones that we'll see and some of the really particularly relevant ones that can really propagate themselves. All right. So the last thing that we need to talk about is the different structures of DNA. So we've been focusing on the B form structure because that's what's most common. So everything that we've talked about so far is a characteristic of the B-form DNA. But DNA can actually adopt other conformations, all right? It can adopt an A conformation or a Z conformation. So let's start with the A form. DNA usually adopts this conformation in dehydrating conditions. Dehydrating conditions. All right, and what are the key characteristics of the A form? It's right-handed, just like B form, but you'll notice in the B form that these bases are nearly perfectly perpendicular to our helical axis. That's not so much the case in A form. All right, so bases are not perfectly perpendicular. To helical axis. Another difference is that there are 11 base pairs per turn instead of the about 10 that we said were in a full turn in B-form DNA. And that means that instead of having that 3.4 angstrom distance in between each base pair, we now have 2.6 angstroms between each base pair. So that is this distance here is going to be that 2.6 angstroms. Right? And one more difference is that, so this is the minor groove in A form. This is the minor groove in B form. So in A form DNA, the minor groove is much wider. And it's also very shallow. So minor groove is wide and shallow. And the major groove is deep and narrow. So those are some of the key characteristics of A form DNA. So now let's talk about Z form. Z form, very unstable. And it's very short-lived because it's very unstable. All right. And this can arise due to specific cellular conditions. And it's also been observed in high salt conditions. All right. And this is the one that's the most different. So to start off, it's a left-handed helix. Right, so that means that the two strands are wrapping around each other in the opposite direction from our other two forms of DNA. There's also 12 base pairs per turn. And it's not as short and condensed as A form DNA. So it's actually much more expanded. So hold on, let's add short and condensed over here. And then expanded over here. So we have 3.7 angstroms in between each base pair. So that is this distance, right? So that's actually higher than it is in B form, right? And then the last kind of key characteristic of Z form DNA is that our major groove and our minor groove look very similar. There's very little difference between the major and minor groove. All right, and so that wraps up our segment on nucleic acids. In the next video, we'll be discussing amino acids.